So let's see if we can. So thanks to the development of new chemical tools, we know today the DNA sequences of a large number of different genomes. But to merely read this information is not sufficient to understand life's inner workings. We also need to have tools so we can change the information and find out about the function. With the discovery of the CRISPR-Cas9 genetic scissors, Emmanuelle, Charpentier and Doudna has provided science with such a tool. We can now change the genetic information in any cell and any organism. And we can find out the function of the genetic material. The discovery of CRISPR-Cas9, the system, comes from studies of ancient immune system present in bacteria and other microorganisms. So just as we can be infected by viruses, so can bacteria. And if they suffer a virus infection and they survive that infection, then they keep a piece of the viral DNA as a memory. And this is done in a specific region called CRISPRs, where you have multiple small memory pieces of DNA. So this is where the information is stored. But how can this piece of DNA actually defend the bacteria from later infections? Well, in order to do that, you need to transcribe these regions. So you need to make a copy of CRISPR DNA and make it into RNA instead, CRISPR RNA. And in the next step, you need to actually take this CRISPR RNA, and you need to sort of cut it up in smaller pieces. So you get small pieces of RNA that correspond to one single infection event in the past. And how